So we'll take a couple of minutes and then we'll begin with our first speaker. Before I give the screen to him, I would like to introduce our first speaker, Dr. Mike Toscano. I am very excited to hear his talk, and he will be presenting his seminar on, and he will discuss and highlight the challenges in breeding laying hens for the cage-free futures, where to ensure high productivity as well as high welfare. Dr. Toscano is a group leader for the Center of Housing for Poultry and Rabbits at the University of Bern in Switzerland. He was born and raised in the United States and finished his PhD from Purdue University, but he has been with the University of Bern since 2013. His research focus mainly lies in keelbone fractures and keelbone issues in laying hens, as well as looking at individual and flock level behavior in cage-free houses. And he also is exploring ways to optimize the housing design, especially feeder spaces, and looking at other problem behaviors such as toe picking and piling. Dr. Toscano is also the section editor at Poultry Science Animal Welfare and Behavior section, and he is a member of PSA, ISAE, and he has attracted significant level of funding from different governmental and non-governmental sources. So Mike, the screen is yours, and then I'm very excited to hear this talk.
Okay. Thank you, Prafula. Thank you to everybody for the introduction and the opportunity to speak today. I'm now going to share my screen. There we go. So I wanted to say thank you to everybody for the opportunity to present today. These are some ideas that have been developing in my group and going in lots of interesting directions. So I'm really keen to get your feedback and to let you know some of the things we're doing. So the title is The Challenges in Breeding Laying Hens for a Cage-Free Future to Ensure Productivity and High Welfare. So I think I should start off in saying that for those that are very keen on the genetic aspects, that's definitely here, but this is very much a talk about breeding and genetics from the perspective of behavior. So hopefully that's not a disappointment. So and about for today, I think that one of the emphases here is that actually it's on the global standards and researchers and research advances in poultry welfare. So I would say that this is an issue because breeding by itself, there's very few breeding companies in the world. Basically outside of China, there's 3. So really what happens in one nation or within one company has global implications for how those things are done. The Lohmann selected leghorns that are present in Switzerland are also going to be present in your nations as well. So really, there's global implication for what we're doing, despite being having very local effects. So as an outline for today's talk, I wanted to talk about the specifics of just an outline for housing transitions. Basically, we're taking birds from cage to cage-free, and then the mismatches in welfare, where we think animals should have high welfare, but they often don't. Then I wanted to discuss breeding as a problem, and then also what I see as a solution, methodological challenges to that breeding effort, and then some solutions I propose for this topic of a grant that we received called HenTrack, and the focus on the individuals in large groups. And then hopefully there's time, we'll talk about the importance of individualized responses and behavioral structure. So first, transitioning. We're now across Europe and also increasingly in the US and other developing nations, we're seeing a transition from cage systems to cage-free. And this is really an important decision because it's, and there are a lot of factors that need to be considered. For instance, just the housing of the animal. To my pointer, the housing of the animal, because the barn is going to be built, mortgages need to be made, it's going to be a 20 to 30 year decision that the producer is going to need to live with. So it can't be taken lightly. Hybrids have to be decided. There are particularly going to be certain behaviors that are going to be associated with particular hybrids, as well as particular egg colors that may be more favored by a part of the population. We need to think about the diet and the feed source and ensuring that those are sustainable over the long term, as well as the health of the animals and the farm size. So these are all important decisions that need to be taken and considered when we're determining what we're going to transition to. And with our focus on cage-free systems, for this talk, I'm going to be primarily focused on the housing and the hybrids that are involved and the decisions that should be made regarding those decisions. So first, most of it will be from the decisions being made are from cage to cage-free. And I'm sure everybody knows what these are, but this would be a typical cage system. And then this would be an aviary system, which is one popular cage-free system. So when you look at the pros and cons of this, cage systems are typically going to have less costs, and they're going to be easier to manage because the animals are retained in these cage systems. Cage-free is generally considered high welfare because the birds have a greater ability to move throughout the entire barn and thus perform natural behaviors. They may also be given access to outside areas. So generally, cage-free systems are considered high welfare because of these benefits. The animals are able to express these natural behaviors and have a greater type of movements available. However, cage-free is often not very welfare friendly, and we have this mismatch between what we would like to occur and the reality of actually what we see. And I'm going to cover three particular issues that I think are most dominant: Keobalon fractures, toe pecking and feather pecking, as well as piling and smothering. So these are three common problems that we see in cage-free systems that I think make them very non-welfare friendly. So Keobalon fractures, this is a bone that's located right here. It's an extension of the sternum. And typically we'll see anywhere from 70 to 80% of a flock will have some level of damage by, say, 50 weeks of age. And these are radiographs of the keel. And you can see on the left side, you have images with no fractures, and then you get progressively more severe fractures all the way to 5. So we would associate this with reduced welfare as well as decreased egg production. We can also see problems in toe pecking. This is something that has appeared very recently, within the last, I would say, 3 to 4 years. We don't really know what's causing it, but it seems to take off in a flock, and then it becomes progressively worse, similar to what we'd say feather pecking, where we'll typically see maybe 50% of a flock with gross aspects of toe damage, where they'll have an entire toe missing or other severe damage. We also see the problem of piling and smothering. And we can see here is often the producer will find, say, anywhere from 3 to 20 birds that appear to have suffocated in a corner or some area of the barn. And it seems to be that even so I'm going to turn off my volume because it keeps. So we often see that this consistently appears in certain locations in the barn for certain herds, and we don't really know why. And this is an image, some images that we've taken from our flock, where you can see as we move along, a group starts to accumulate here with more and more birds that are piled together. And if this is not dissipated, we seem to see that they smothered and can actually be killed. So these are just 3 problems that we see, and these all are, I would say, relatively common in cage-free systems. And I think if you look at that, it's definitely not welfare friendly. So this is, sorry, I'm having some technical issues there. So this is definitely something that reduces the welfare aspects for cage-free systems, and it's something we need to solve. So when we consider the motivations to move from cage to cage-free, there are many reasons to not use cage-free systems. Cage systems, like I said before, are going to be easier to manage. They're often simpler to clean between and disinfect between the flocks, and we have less behavioral problems. But at the end, they're still cages. But the reason that we would like to move away from cage systems towards cage-free is because of such limited movement and such limited ability to perform natural behaviors. So despite these problems, the alternative is cage systems, which we believe is not a very good option. So we really need to improve cage-free housing. So now how can we do that? Well, we can work to optimize cage-free housing and say, work on specific problems to alleviate them. So one example, this is with Keobalon Fractures, and we've tried to implement ramps. So say if you take an aviary system, and this bird could be, say, 3 meters above the ground compared to here. And to say this bird wants to get to the ground, instead of flying down, which is often associated with a collision and damage, the bird can walk down using these ramps. So we've been able to show that reduces Keobalon fractures by about 20 to 30%. And we have lots of research that are involved now actually training the birds when they're very young during the rearing phase to actually learn how to use the ramps. And these are some images within our systems. So this is definitely one way that we can improve cage-free housing. But to use a phrase that's common for the native English speakers, this is a bit of a whack-a-mole problem where you solve the symptom, but you don't really solve the underlying problem. So as soon as you solve one problem, another one pops up. So we're constantly trying to correct these problems without actually treating, So we're constantly trying to solve the symptoms, but never really dealing with the underlying problem. So this is where I suggest that actually part of the problem is breeding and that we are not breeding correctly. So the way that we breed right now is based on small groups of birds in cages. And this is done for several reasons. The ease and accurate ability to generate a phenotype, where a phenotype is defined as the observable trait, something like eye color. So within small cages, say I can look at this bird and I can easily identify, say, its feather coverage or some other trait of interest. I'm also going to minimally disturb the animals. I can look at the animal easily and I can observe the traits that I need without causing a lot of disturbance that causes stress to the animals that would affect its performance. And also these methods, because they're in cage systems, the vast majority of the world maintains their laying hens in cage systems. So for all of these reasons, the current methods work very well for cage systems. However, for cage-free systems, this can be a problem. In cage systems, we actually do see fractures, but they tend to be very, very small and less severe. We rarely see feather and toe pecking simply because it's often contained within a particular cage and is not able to spread throughout the flock. And similarly for piling and smothering, because you only have a small group of birds, say maybe 8 birds or maybe 50 birds in a furnished cage, you're not really going to get these large smothering aspects. So we actually can't phenotype and actually observe the traits that we see that are problems in cage-free systems. And if you can't observe it, you can't breed against it. So what we really need to do is, instead of breeding for these types of systems, we need to think about the animals in these types of environments. So if I want to, say, select for this particular bird here and the phenotypes that this bird has, I need to be able to quantify those traits. I need to be able to see that trait. But if the bird is mixed throughout the entire flock of these massive systems, it's very difficult to see. So I wanted to speak about specific relevant differences between the cage and cage-free systems. And I'm going to focus on 2 aspects. First, social structure, and then animal movement. By the way, if there are any questions that are coming up, The moderator should just ask me to pause, and I can explain something in any more detail that's needed. So firstly, social structures. Okay, I mentioned before we have small groups in cages versus large cage-free groups where we may have thousands or tens of thousands of animals. And we're going to see very different social structures within these two settings. So this is some excellent work by Leith and Keeling from a 2003 paper, where they looked at social structures and the amount of agonistic interactions between small groups of animals that were housed with 10 individuals or large groups with 120 individuals. So they had animals in these two different settings, and they had 8 pens of small groups and 8 pens of large groups. And then what they would do is they would take an animal from, or 2 animals from the small groups, and they pair them together in a small crate, and then they would observe their interactions. They would do that for small groups, and then they would also take 2 animals from the large groups, either animals that were from the same pen or different pens. And then they would look at the fights that occurred. In the small groups, out of 128 pairings, they saw 48 fights. Whereas in the large groups, out of 128 pairings, they saw 48 fights. And out of the large groups for 128 pairings, they saw only 3 fights. So a dramatically increased number of fights in the small groups, animals that house in the small groups. And these graphs, you can see this is a number of tests performed. And on the x-axis, you can see the day of test, where the dark bars represent a test that resulted in a fight or aggressive pecking of the light bars. So you can see here that in small groups, these animals were much more aggressive. They were primed to actually have a very different social structure and interact with the animals very differently. So what explains this? This is a concept that was suggested by Estevez et al. In 1997, where it's the tolerance hypothesis that indicates that it is not efficient for birds housed with unlimited food and water to spend time and energy defending resources from other individuals when the number of competitors is high, but competition has little effect on the depletion of resources. Basically, in small groups of birds, it makes sense because the animals have a limited number of resources, and because they can recognize all the other individuals in the group, it makes sense for them to be aggressive and to establish a hierarchy. But in large groups, when there's unlimited amounts of these resources available, and also it's very difficult, say, with 1,000 birds or 10,000 birds, to recognize the animals that you've had fighting encounters with, it would be very difficult to establish that hierarchy. So it doesn't make sense to fight. However, we can see that there are different resources within cage-free systems that may be limited. This is anecdotal, but when I was in the UK and the birds would have access to outside from the pop hole, we'd often see birds that would spend massive amounts of time in the pop hole. And we believe that was maybe a resource that the birds were protecting. So they have unlimited feed and water, but here they may be protecting this particular resource. So there's very different situations in between cage and cage-free that we need to account for. Another thing we need to think about is housing differences, and specifically how the animals move throughout that system. So if you can imagine that this particular bird here can access anywhere in this barn, whereas a cage system, it's maintained its position in a very small space. So the bird actually has to navigate through this entire area. It needs to learn, say, where is the nest box, and also how does it get there. If it wants to be in the litter at one moment and then roost higher at another point, it has to learn how to move throughout that environment. So as I mentioned before, we can install ramps here, but then we even need to teach the bird how to use these things. And there may be different skill sets that are required there. So there are very different requirements for an animal that lives in a cage system versus a cage-free system. And we need to breed animals that will be able to manage these differences. So this is where I see breeding as being part of the problem, but I'm also seeing it being part of the solution. And this was a project that we're beginning. We're still organizing it now, but it's set to begin in the next 3 months, called Hen Track, where selection is going to reflect the cage-free environments. And as I mentioned before, these are going to be very different social structures, resource availability, and motivations that we're hoping to include in these selection paradigms. So I just wanted to cover some of the logistics. I'm very lucky here to have a facility where we actually have a vencomatic bow-leg terrace aviary. That this is an overview of the system, a bird's eye view, and this is a side view. And this is a commercial operation, like many you would see if you went into a commercial barn. But what our system is, it's divided into 20 pens, where each pen contains 225 hens per pen. And for this particular project, we're focusing on 5 pens for a total of 1,125 hens. So we're going to be monitoring the behavior and the various traits that we're interested in these pens, while our industrial partner, Hendrix Genetics, is going to be also doing the evaluations at the individual level for egg yield and feed efficiency. And by combining that data, the idea is that we can actually breed a hen that will perform better in the cage-free environment while also in a cage environment. So just because I threw out a lot of information, I thought I should stop right here and just make certain that everybody's caught up. So I've talked about housing transitions and that we're gradually moving from cage to cage-free, largely because we believe cage-free systems are better welfare. However, there's often a mismatch here, where there's many problems that we see in cage-free systems that do not make them welfare friendly. And this is a major problem. I think part of this problem is rooted in breeding, specifically that we're breeding for the wrong environment. But I also see breeding as a very important solution to help resolve these problems so that cage-free systems can indeed be high welfare. So hopefully you're caught up. So when we're breeding for cage-free systems, there are going to be several things that are required for the evaluation of these traits. First, we need an accurate phenotyping of the individual. We need to actually collect that information at the individual level. We need to use conditions that are relevant to the intended environment. So we need to actually evaluate these individuals with an environment that's cage-free and large groups, typically in the thousands. At a minimum, we believe the social structure is very different above 100 animals. So ideally, we want to focus on more than 100, but from a genetic standpoint, you really need to use thousands of animals. We also want these evaluations to occur where they cause a minimum disturbance upon the animals. As soon as you disturb the animal, you're going to affect their productivity. So we need to minimize those disturbances. So as we look at this, there's several methodological challenges. Specifically, I see this for genetic selection, but more broadly for animal welfare scientists. For all of you that are involved in improving the welfare of laying hens in cage-free systems, these are also challenges for you as well. So typically, we can do individual level observations on traits like egg production, aggression, Keobalon fractures, or personality. So all of these things, I can look at this individual in the cage. I can see how many eggs it's laid. I can see if it has a Keobalon fracture. I can assess its personality. And I can do all that quite easily, either in the cage or removing the animal out and placing it in some type of a test chamber for personality. But that's going to be really different when we have these large-scale cage-free systems. How can I observe the individual here? How can I observe an individual reliably over multiple time points? It's very difficult to do that. So how do we assess the individual, but within large groups? So in the facility that I mentioned, where we have our barn on site, where we have 225 birds in the pen, we will routinely do health assessments on those birds. So we will routinely assess them for feather coverage, foot pad and leg condition, as well as the Keobalon fractures using radiographs and palpations. So with this, we can get an accurate phenotype, and because it's occurring in a commercial barn, we can combine pens as well. So we can have upwards of 225 even to 1,000 animals per pen. So we're doing these health assessments in conditions that are relevant to our target environment. However, we're still going to cause these disturbances because we have to actually go into the pen and grab that animal to actually see what the feather quality is. Does it have a Keobalon fracture? We're still disturbing the animal. I also wanted to mention that I think this is really important when we're focusing on these large-scale commercial facilities, that we should take advantage of them that are now increasingly around the world. The Aviaforum, this is a facility that I'm based at in Switzerland, but also the Experimental Poultry Center in Belgium, also the Poultry Teaching and Research Center at MSU in the United States, as well as in commercial partners. So I think when doing these types of assessments, we need to increasingly be thinking about how we can do these assessments in these large-scale commercial facilities to ensure that our findings are relevant for actual commercial poultry production. So I warned earlier that I'm not a geneticist, and I should also warn that I'm not a technology expert. A lot of the things that I talk about, where I talk about the use of technology in the next few slides, I should say that we use a lot of technology, but I benefit by having really good partners that can help me with the technology. So if there are questions about the specifics of the technology, I will do my best to answer them, but often this is a topic for somebody else. So our goal is to minimize disturbance of the animal, and I think that we can do this with remote tracking and use of technology. And I'm going to give you several examples for how we do this and how it's revealed some very interesting ways that we can examine these traits. So this is our system in the barn, where this is the bow-leg terrace aviary that I mentioned before from Vencomatic. And what we've done is we've divided the barn into 5 zones. So zone 5 is the highest tier, zone 4 the middle tier, 3 is the lowest tier here, and zone 2 is the floor, and then finally the winter garden. And each of these zones have specific resources that we know are very important to the bird. So zone 5 has feed, water, and perches, and it's where the bird will roost in the nighttime. Zone 4 has nest box and also perches. So simply, if a bird wants to dust bathe or litter scratch, it's going to need to go to, say, zone 2 or zone 1, because that's where the litter is. If it wants to lay an egg, ideally it's going to use the nest box, which is located in zone 4. And what we can do is we can track animals as they're moving throughout these 5 specific zones. We don't know where the animal is within a zone, but all we know is that it's in zone 5. And on this graph here, you have a time series plot. Say we're on the y-axis, you have the 5 zones represented, 5, 4, 3, 2, 1. And on the x-axis, you have the time of day. And this is the plot for, say, one animal as it moves throughout that. So what we saw when we began tracking is very subtle differences across and within animals. So this is a plot that I mentioned before. And what this is actually a plot, a time series plot for one animal on multiple days. So this would be animal 1 on day 1. And you've got the 5 zones on the y-axis. And then on the x-axis, you have time with lights coming on at 2 and then going off at 5:30. So this particular line here is 1 day. This is the 2nd day, 3rd day, 4, 5. So each of these graphs or each of these lines represents a day in the life of the animal. And what you can see is for animal 1, you see a very consistent pattern, day after day after day. And we thought that was very interesting. But what was even more interesting is when we looked at other animals. So this would be animal 12 and animal 37, say 2 different animals, but in 2 different animals on the same day and in the same pen, but they had a very different pattern. So you can see animal 12 has a very different from animal 1, and 37 is also very different. But within an animal, we saw lots of consistency. So we thought this was very interesting from a genetic perspective. And we began examining like how we could actually quantify these differences. So this gave to this term that I've been trying to develop, which I call behavioral structure, whereas a consistency of movement and location patterns, or MLPs, that are observed over extended periods of time, such as weeks or days, with high resolution data. Now, I should say this behavioral structure is something that others could use and others have used. But what's really unique about what we're doing here is that we're able to generate high resolution data. So whereas we typically say would observe the animal's location, say at 5 time points over during the day, using something like video observations, we would get a picture and say, so we'd have 5 time points on the animal. But now with technology like this, we can actually record the animal's location, get a recording basically every second of the animal's life. And we can do that for months and months on end. So it gives us a much richer picture of the animal. And I think we can use this data now to develop traits that we can now breed for and to give us indicators that are really important for the animal's welfare and production. So to remind you, I said that goals for trait evaluation needed to involve accurate phenotyping as well as being relevant to the environment. And now what we're focusing on is, well, how can we do this by minimally disturbing the animal? So one way that we could use this data is to look at as an indicator for keel fractures. And what you can see here on the y-axis, this is the duration of the time the animal stayed in zone 5, which is the top tier in minutes. And on the x-axis, you have increasing fracture severity. And what we see here is that as fracture severity increased, the animal spent more time in the top tier. So again, instead of actually grabbing the animal and having to do a radiograph to determine if it has a fracture, we can essentially use remotely collected data. And if that movement data and where it's spending its time is associated with keel fractures, we can select for that. We also use techniques for classifying the data regarding variation over time. And this is this concept of entropy, where we see actually after vaccination, we see a rapid drop in the entropy. And here in these graphs, you can actually see low versus high entropy. And this looks at actually each day, you can see a fairly consistent movement and location pattern. So this day is the animal's performance. This day is very consistent to this day and very consistent to that day. We would say this has low entropy compared with this animal, which has a very different pattern each day. We would say this animal has high entropy. And what we've seen that following a period of vaccination, we see that entropy has a rapid fall. So we can actually, and then we can actually see that the animal begins to return to a normal pattern. So the idea here is that we can actually see if how quickly does it take for animals to return as an indicator of health. And then we can actually select for that rather than grabbing the animal and say taking blood samples or other measurements to determine how effective the vaccine was. We also see patterns that seem to be very much grounded in physiological mechanisms. So when you look at this movement and location pattern, if you specifically focus at zone 4, where the nest box is, and over the day, you can actually see a slight shift in when the animal is accessing that zone. We think that is probably most likely associated with the animals accessing the nest box to perform egg laying. So these behavioral patterns and the shifts that we see seem to be very much focused on physiological control mechanisms. Okay, so focusing on that, what's been really interesting about this is that the more we look, the more that we find. This is a paper that was published in Comparative Biochemistry and Physiology, examining this concept of behavioral structure and epigenetics. And for this, we actually looked at animals. This is in a different barn where the animals could go outside, and they had access to a barn area indoor. They could then go access out to a covered winter garden. Then they could access to a yard that was covered in stones, and then finally they can move out to the free range area. And what we did is we observed animals as they moved through these locations, and we examined their epigenetic profiles. So epigenetics is another thing that actually I'm not experienced with, but just to give you a quick primer of it, we all have DNA, and every single cell that we have in our DNA, every single cell that we have in our body has the same DNA. And that DNA will be the same in every cell throughout our entire life. However, when there are certain parts of your DNA that will have these DNA methylations. And what that allows is particular areas of the genetic, of your DNA to be enhanced or inhibited. So certain parts can be expressed more or less. And that is actually your epigenome. And every single cell in your body, in theory, has its own epigenome. And we actually see that this contributes a lot to the variation in the phenotypes that we see. So for this study that I mentioned, we have 4 pens that use 365 Lohmann Browns per pen. And of the 365, we tracked 120 per pen. And we tracked the animals for 4 months. So for 4 months, we could actually know where the animal was in terms of the barn, winter garden, yard, or free range area. So when we examined the brain, specifically the nidopallium at 42 weeks of age, and we compared animals that either had low entropy over the period of time versus high entropy, we actually saw a very different epigenetic profile. So this on the x-axis is whether animals were classified as low entropy or high entropy. And each of these bands is a specific region of the genetic area that was either inhibited or enhanced, where darker red is more inhibition. So we see very different profiles in how we classify the animals. We also see really big differences in terms of the epigenome with how we classify the behavioral structure. So let me give you a couple of examples. For one, we actually have this concept of coordinated movement, where we could actually, when an animal left an area, we could see whether other animals followed that same path within a 5-second interval. So we could look at this coordinated movement, which is largely an indicator of sociality. And we could also determine whether certain animals were leaders, so they were always going, or whether animals were following a particular animal. So am I going and I'm with her? So we could actually classify animals as leaders or followers. We could also classify animals using a concept called dynamic time warping, which examines the whole entire pattern and classifies the animals based upon the time series. And by looking at that, we can actually see 3 clusters of animals. So in this cluster 1, you can see animals access all areas routinely, but rarely spend much time in the outside areas. Cluster 2, you see much more transitions constantly throughout the day. And then cluster 3, the animals pretty much spend their entire time inside the barn. So now what's interesting here is that dynamic time warping, when it classifies an animal, the animal is going to be classified this way and remain this way over its entire lifetime. So it's going to be fairly rigid. Whereas the social index, particularly for the follower animal, it's going to be very flexible. An animal in, say, cluster 1 is always going to adapt this profile. Whereas a follower animal, it's going to depend upon what the leader is going to do. And when we looked at the different epigenetic profiles and how we classify these animals, we saw different regions of the DNA that were affected. So if we look at animals using the sociality index, we see 110 areas that were either inhibited or enhanced. And 107 of those were unique, whereas 1, there was an overlap with entropy, and 2 was a dynamic time warping. So depending on how we classify the animals, we see very different regions of the epigenome that are responsive. And we think that this is largely associated with how these, what traits that these classification schemes are relating to. So to summarize the genetic evaluations, we need to be doing these evaluations within large groups of a commercial system if they're going to be relevant to make certain that we can deliver genetics for cage-free systems. We need to assess the relevant traits of interest, including health and injury, and we should use remote tracking of individuals so we can minimize disturbances to the animals. We can look at patterns and routines and link them to standard traits, and we can collect novel health indicators remotely. I think we should also use this concept of behavioral structure over time and across animals as a part of this genetic evaluation. So in conclusion, cage-free must be optimized with better breeding, where we need to include selection in relevant environments, healthy animals, and high performance. Otherwise, cage-free systems are not going to deliver the welfare that we need. And we should use traditional and alternative evaluations, targeting mechanisms with a strong biological basis. So I just want to give lots of thanks to members of my group, ZKG and helpers, as well as many collaborators and partners, and also many funders who supported this work over the years. So with that, I can take any questions.
Thank you, Mike. And we have 2 questions for you. So I'll start with, is smothering positively associated with fearfulness and anxiousness in a flock? Or have you noticed any of that?
Sorry, could you repeat the question again?
Is smothering in cage-free associated with the fearfulness or anxiousness of the flock?
No, that's quite interesting. So we often see, no, actually I would say no. We often see piles accumulate quite passively, where the animals often they're attracted to a particular stimulus and they gradually become attracted to that particular area. We've also, this is just again anecdotal, but we've seen that some animals will actually see the pile develop and rush towards it. So no, we don't think it's that there's a panic that's the animals are scared into that. We think that's something different. The animals seem to actually want to go there.
Yeah, it was interesting because there were a couple of reports that were saying like some of these situations where birds are under stress, it actually promotes the risk-taking behavior, and they try to make a bigger jump or go towards these scenarios where they're taking bigger risks. Maybe something there. Okay, so another question is from Greg Fraley. Sorry, I missed this, but in regards to leaders and followers,
Yeah.
how consistent are these classifications from day to day? Are the followers following the same leader over time? Are there consistent cliques or microgroups that interact with within a barn?
Yeah, so actually, I should say that when we did this study, we really didn't expect to find what we found. We just expected there'd be some animals that ranged and some animals that didn't range. So we developed these methods almost after the fact because we realized this was occurring. So I should say now we know much better what to look for, and we can actually plan this much better. But that's a really good question. We actually did see that the structure would actually become more consistent over time. So say if there was a particular follower leader structure or combination, we would actually see that over the 4 months, that became more consistent over time. So if anything, it seemed like the groups tended to actually become more stable. So yeah, I think that answers your question.
So there is another question. So in terms of genetically selecting for cage-free environments, would selecting for physical attributes that reduce the amount of flight or flapping from the birds be possible, such as adjusting their wing loading? What are your thoughts on that? Wing assisted include running would still get them to their resources with ramps.
So maybe I missed that question, but is it breeding for physical characteristics?
Yeah, so breeding for physical attributes that would reduce the amount of wing flapping or flights. So they want, so ideally selecting birds for walking or running more rather than jumping or flying.
Yeah, but yeah, this is actually a really good question. The geneticists really have told me they can select for anything as long as they can see it. So, and I would say that actually birds are not good flyers. And we know that from well, chickens are not good flyers. And when we provided them the ramps, particularly in rearing, they seem to actually very much prefer that. We have a study that's coming out where birds that were provided ramps, 80, even at 16 weeks of age, 80% of all transitions were done with the ramps. So I would say actually that we should be selecting for birds that use ramps and what kind of traits are needed there. So during rearing, they need to develop the cognitive abilities to judge the incline, the musculature. But yeah, so without a doubt, the birds are not good flyers, but they're walkers. So we should be breeding them to help them be better walkers. So I think in terms of the questions you're asking, yes, physical characteristics, but do we want to breed for birds that fly or do we want to for breeds that walk? I would say we should be breeding more for birds that walk.
So there is another question from Kate. What is the heritability for different behaviors that you're evaluating? For other poultry geneticists that find heritability of specific behavioral traits is very difficult and thus challenging to select for this.
Yeah, so you're definitely getting into questions that are extending past my knowledge, but I will say that these are traits we haven't been able to quantify. All of these studies that I've shown here were done with, say, maybe 100, well, I think at most 400 animals. But with HenTrack going forward, we'll actually have the daughter sires. So we can actually trace back to the particular father. So we have more genetic material, and we also have more numbers, where we'll have 1,100 birds. So for these traits, that if we see something that we want to select for, like a bird that when it's vaccinated, it performs, it returns to a more natural behavioral performance. Because we have the numbers, And because we have the pedigree, and we'll also be doing genomic evaluations, we will be able to calculate the heritabilities. But as of right now, that's an excellent question. We don't know the heritabilities for many of these traits.
So with regards to that vaccination, so you said the complexity of the behaviors decrease, or the entropy decreases, but was that true for the low entropy animals as well? Would they decrease even further, or just on the high entropy birds?
Oh, I see what you're saying. No, so I guess the question is, did we see entropy increase in both birds were classified as low and high? Yeah, I would say because of that particular system, because there's only 5 zones, there was a bit of a, I guess, a flooring effect. So there may be further decreases in entropy, but it's difficult to detect. This is, I guess, another benefit of the hen track work, is that we're actually going to have more resolution in how we track birds. So we should be able to do that more. But yeah, that's an excellent question. We don't actually know, did it go further?
Okay, so there is another question, I think, from LG on the same vein. So we're not letting you go, Mike, yet. So the next question was the entropy of the movement associated with the social status, or in other words, how you determine the social status of those birds?
I guess, so this is another question that we just can't answer. I mean, this is what really is annoying presenting this, because so much of the study that we published, whether it was the tracking the birds inside the barn or outside the barn, we didn't expect this kind of data, so we didn't know how to measure it. Now the grants that I've gotten the last 2 years, we've actually, we can now look at this. And this is actually one study that we're looking at. We're actually trying to see the specific social structure, the particular, how a bird interacts with its flock mates in terms of submissive and dominance. How is that associated with these MLPs? But as of right now, I wouldn't be able to give that an authoritative answer. We're very curious to, stop me if I'm not answering the question, but we're really curious to see if an animal actually is very dominant. Can it go anywhere it wants? So that it would actually access all areas and be in a, have a very rigid pattern? Or would we actually expect that if the animal is very dominant, it doesn't need to go anywhere, so it can just stay in its favorite location? So I think these are all questions that we can look at. And from a scientific perspective, we have the tools to do it, and now we know that these patterns exist, so we can do that. So we're planning on answering those questions in the next, say, 3 to 4 years.
So there is another question from Hector Lavia. Do you think that through nutrition you could modify or manipulate some of the behaviors that you study in favor or against your selection?
So I missed the first one. Did you say based on nutrition?
Yeah, so yeah, could nutrition be a factor in promoting or decreasing the traits that you're selecting for in case?
Oh, yeah, without a doubt. And again, I think this is, if you look at, say, some of the graphs where we saw animals that were, so if you see an animal that actually spends almost all of its time in one location. So let's go here. So if you were to look at, say, these 2 animals, this animal goes from the bottom of the floor to the top 13, 14 times a day. Whereas this animal actually seems to stay pretty regularly on one location. So I would imagine that this animal, number 37, has a very different energetic profile than, say, number 12. Now, so yeah, so I would think actually their nutritional requirements can be very different. Do we need to breed for that? Do we need, is animal 37, is it more productive? Is its feed efficiency different, say, than animal 12? These are kind of questions we need to ask. Are their egg profiles different? So yes, but I would say it's very likely that animal 37 and animal 12 and animal 1 have very different nutritional needs.
So yeah, and then just adding to what Mike said, when we were looking at birds using accelerometers for the movement in case-free environment, the birds that moves quite a lot, is associated with a different level of fat in the abdomen compared to the birds that doesn't move a lot. So that definitely the feed formulation for cage-free systems has to be reevaluated. But I guess the thing is to figure out how we do not overfeed the lazy birds and how we do not underfeed the very active birds, right?
And I would say that I think what's really interesting is that we knew there's variation in these birds, right? That should not come as a surprise to anyone. And we could eat, but you can't quantify that in a cage. So, okay, we know that it exists in a cage-free system, but how do you quantify it? So now I think what's really interesting is we know it exists and we have the tools that we can measure this. So it just opens up this whole world of ability to develop phenotypes that we previously could not access. So that's what I think is really so exciting about these opportunities. We know they exist. We were previously limited by our ability to see them, but now we can quantify that, and we can do it in a really robust way.
So there are a couple more questions, Mike. What do you think about cage-free monitoring technologies using drones? Do you think blade noise will increase stress and produce negative results? Have you seen any of these studies?
Sorry, could you say it again? Did you something about monitoring with drones?
Yeah, so could drone be an technology that could be used to monitor behaviors in cage-free houses?
Yeah, I think that'd be great. I would say though yes, but I would say for all, and this is where I am not the technology expert, but the cost of a single bird is going to be very small. So I really think if any technology that we do on a really widespread fashion, say on every farm, it'll be difficult to do that at the individual level, but something like drones or even basic video capture technology at the group level is going to be really important to help us identify problems. So a really simple example, we have cameras installed in the barns and we can see when piling occurred. And to cause some type of an intervention. So I think that, but that's a different question. In terms of focusing on the individual level observations, I really see it only working in certain conditions.
So one question from Allison, are there plans to evaluate behavioral traits in the breeder flocks? Could you discuss the trade-offs of evaluating layer flock versus the breeder flock for informing genetic selection? Because the layer that I think we use in commercial systems are four-way crosses. So how effective would the data be that we're collecting from four-way crosses to apply it for selection in a pure line genetic?
Yeah, sorry, I should have phrased that better. The work that we're doing in HenTrack is not the commercial hybrid. It's actually step above that. It's a step above into the lateral. We call it with the genetics, we call it the elite crossbreds. Yep. But basically the traits that we observe, they can directly link that to the sires in the parental flock. And so the birds that we're using are not going to be what you're going to have in your commercial flock. We can actually trace that back to the pedigree. So that's what's really important about the HenTrack study.
So there was one question about when would we get the recording for this seminar, but only after you have all of these seminars in person today, and then we're only going to send you the recording, but which is, of course, I'm joking, we'll send that to you on a later date, maybe a week from now.
I'm definitely not the technology expert there, so.
And then there is one last question for you, Mike. Could the use of environmental enrichment prevent or reduce behavioral problems like toe pecking?
Yeah, so I guess what's really interesting about our group is that we see these problems and we don't know what's causing it. So we just have reports from farmers that are coming in and they're seeing this problem, but they don't know what's caused, and they're otherwise very good producers. So yeah, my approach to these types of problems is that we first need to see how it happens. Is it actually a bird pecking at itself? Or is it other birds, like with feather pecking, where we believe there's a certain small proportion of the population that begins it? And we think it's a combination of the two, where we see actually birds that start pecking at themselves, that create a little bit of a wound, and then that attracts other birds, and then similar to feather pecking, it's a bit of a snowball. So yeah, I'm not certain if environmental enrichment is going to cause this problem. There are some hypotheses that it actually has to do with the quality of their nutrition, that it's the birds are actually developing dry skin or possibly have some type of fungus. But based upon what we've seen, I'm not really certain if enrichment would work. Just before you do cut me off and go on to the next person, I just wanted to, so if you could give me a 30 seconds.
Yeah, you can take a couple of minutes, Mike.
Okay. I just wanted to say I really appreciate the opportunity to speak today. This is a very fresh concept. I'm definitely not the geneticist. When I do speak with my genetic colleague, they often laugh and say, "No, Mike, you got that term wrong, and no, that's not the sire pedigrees and other things." But I do hope that people, I'm very eager to hear feedback from people. So please contact me through the website. If you'd like more information, I'd be happy to explain it to you. And like I said, I think this is research being done in Switzerland, but I see it having very global implications. So I'm very, very, very keen to clarify if there is any confusion and to get feedback from people that would like to give it. So thank you very much, and thanks for the opportunity to present.
We thank you, Mike, for the very exciting talk and fostering this discussion. So let's wrap your presentation here, and then I'll hand over to Leonie, who would introduce our next speaker. Thank you again.
Okay, and then I have to stop sharing now, don't I? Yes, there you go.
Yeah, so thank you, Mike. That was